We are clear on where different pumps and channels are located within the neuron. Yes? Because we're going to talk about the different parts of the neurons today and what they do. Okay? In terms of an action potential, either initiating, receiving the signal from another neuron, initiating an action potential, and then conducting the action potential down the axon. All right, so if you are not clear on where the types of pumps and channels are in the neuron, you will be confused as we talk about today. All right, so what types of channels are present in the receptive parts of the neuron? Neely. Chemically gated channels. Okay, so we're not going to have chemically and voltage. They're going to be separate, but they're chemically gated. Okay, that is absolutely important. Okay, so first off, we're going to go through. I just went through the review of the different parts of the neuron: receptive segment, initial segment, conductive segment, and then the axon terminus. We're going to talk about how those segments, how the different parts and the channels that are associated with those initiate and propagate an action potential today. And we'll compare, we'll talk about what a graded potential is versus an action potential, because there are two different ones, okay? And then we'll actually go over how different factors influence the speed of conduction, right? So neurons don't propagate an action potential at the same rate, right? They can go faster or slower depending upon what type of neuron it is, some characteristics of it. And we'll talk about those today. Okay? So first, we just said that chemically gated channels are present on the receptive segment. What part of a neuron is the receptive segment? So here's my lovely multipolar neuron. Which, A, B, C, or D, which part is the receptive segment? A. The dendrites and the cell body are where the receptive segment is. Okay? So, this particular neuron is going to be a target for other neurons. Okay? So, another neuron will come in and end here. And another... I can draw a bunch of different ones, right? So a bunch of neurons will terminate on this particular neuron. Does that make sense? The blue neuron is going to receive signals from the other red neurons. Does that make sense? Those red neurons are what we call presynaptic. Pre means before the synapse. Okay? The blue neuron is your postsynaptic neuron. Clear? So, the red neurons are releasing neurotransmitters, are releasing chemicals that will target receptors on the receiving segment of the postsynaptic neuron. Is that clear? All right. So it releases the signal. Thank you, sir. It releases the neurotransmitter, and the neurotransmitter binds to its receptor. What happens? So let's draw it in. Let's draw it right. We're here. Here's our lovely axon terminus. Here's the dendrite of the postsynaptic neuron. Okay? We're going to release the neurotransmitter into the synapse, and that's going to bind to our receptor. Okay, it binds. What happens? That's a channel. The channel opens. And depending upon what channel that is, right, let's say it's a sodium channel. Which way is sodium going to move? Into the cell. Say it's sodium, it's going to go in. What if it's, okay, sodium goes in. Let's say this channel, let's actually draw it in a different color so we see differently. Let's say this particular neuron, that receptor, binds. Let's say the yellow is a potassium channel. Which way is potassium going to move? Nate? Out. Make sense? Okay. Let's say that neuron has a receptor for chlorine. Binds. Which way chlorine is going to move? Do you remember? Look back at the lecture from Monday. Which way is the chlorine ion going to move? Where is it? In. All right, so sodium's a positive ion. Potassium's a positive ion. Chlorine is a negative ion. What does this have to do with anything right now? Think about what the charges and the way they're moving. What do we know about the difference between outside and inside of this targeted neuron at rest? Molly?
It's negative 90.
It's negative 70. Right? For neurons, it's negative 70. For muscle, it's minus 90. All right? So it's minus 70 millivolts at rest. Right? That's our resting membrane potential. Yes? All right. So let's say this neurotransmitter binds to the sodium, binds to its receptor, and that receptor is a sodium channel, right? And a positive ion goes in. What's going to happen? What's going to happen to our resting membrane potential? It becomes less negative. It goes from minus 70 to minus 65. Does that make sense? Okay. Let's say that's a different neurotransmitter binds to the potassium ion channel. What's going to happen? Potassium's going to leave. What's going to happen to our resting membrane potential? It's going to go from minus 70 to what?
Minus 75.
Is it going to become more negative? Yes. It's losing a positive ion, so it's going to become more negative, right? So it's going to become, for potassium, it'll lose to minus 75. What about for chlorine? It's going to become less negative, right? It's going to move up to minus. I'm sorry. You were right. I'm sorry. Minus is going to become more negative. It's adding a negative, and it's going to become more negative. And what does that mean when it becomes more negative? It moves further away from threshold. Okay? And as it moves, anything that moves it further away from thresholds is what we call hyperpolarization. Okay? Hyper means more or higher. It's higher polarized. It's further away from threshold. Sodium is the only one that's going to move it closer to threshold, right? And technically, that is a depolarization, but haven't reached threshold to form the depolarization part of an action potential. Okay? So on our lovely graph of an action potential, if we do it out, right? So we would do it like this, and this would be our minus 55, our resting membrane potential, our threshold, and we're at resting membrane potential at minus 70. Okay? And so a sodium moves in, what happens? We start to go up. Does that make sense? And another sodium moves in, it goes up. Another sodium goes in, it moves up. And, let's say after that, a chlorine comes in. As the chlorine comes in, guess what? It goes back down. Okay? These small changes in resting membrane potential are called graded potentials. Does that make sense? They're graded. They are not, there's different values to them, different we call magnitudes, okay? And they are less than threshold. And they can move both towards threshold, they become depolarizing, or they can be hyperpolarizing, moving further away from threshold. Is that clear? That's what this first slide's all about. Okay, so here we talk about the change and a graded potential that can vary in size. Okay, it is a local potential. It only happens in that part of the membrane. Does that make sense? A local potential is also another example of a local potential are the potentials in the motor end plate or the end plate potentials, right? So before in a muscle, a motor neuron releases acetylcholine. Acetylcholine binds to its receptor, opens the channel, sodium starts to move in. Before that increase in sodium reaches threshold, that is considered a local potential. And once it reaches threshold, then it can spread to the rest of the muscle cell. Then it becomes an action potential. Does that make sense? Same thing happens here in the neuron, right? Local potentials, local changes in voltage, right, are kept around where the neuron is in the receptive segment until all of the signals, right, from all of the input neurons are added together and determine whether it reaches threshold or not. If it reaches threshold, then you get the production of an action potential. Okay? And so this part, the direction of the potential depends on what type of ion. Right? We just went over this. If sodium channel's open, right, becomes less negative. If chlorine channel's open, becomes more negative. And then also if potassium channel's open, becomes more negative. That movement of ions is dependent upon the concentration gradient alone, right? So sodium is higher outside the cell, chlorine is higher outside the cell, and potassium is higher inside the cell. And then you can talk about what hyperpolarization is. Questions on graded potentials? So how are graded potentials used in the impulse generation of a neuron? Typically, neurons do not have just one input. Neurons are targeted by dozens, hundreds of other neurons. Okay? So they're not alone in space. All of these other neurons terminate on a particular neuron we're looking at. And at any given time, some of those neurons will be producing signals and trying to get the target neuron or the postsynaptic neuron to generate an impulse. Or the opposite, trying to prevent that postsynaptic neuron from generating an impulse. Okay? So postsynaptic potentials, right? Those potentials, those gradient potentials that we see in the target neuron can be what we call excitatory or inhibitory. Inhibitory means to counteract. Excitatory means to stimulate. So an EPSP, an excitatory postsynaptic means the target neuron and the potential, right? The potential is just the change in electrical voltage. So the presynaptic neurons here are releasing the neurotransmitter, binding to the receptor, right? And sodium is moving in. Sodium is going to make the resting membrane potential become less negative, right? It's going to move closer to the threshold. That's what we see here in this graph, right? That's what we see here. The stimulus, and EPSP, we're getting closer to threshold. Yes? What would happen to this graph? What would happen to this graph if a chlorine ion was open? A chlorine ion channel was open. We'll do it in red and say it's right here, right? So we're at resting membrane potential. What would happen? Great. Anyone? It would go down and be less negative, right? So we'd have that dip instead. Questions? And that's what you see here. Okay? Oh, where we go? So, the stimulus, this could be a potassium channel opens, so we're losing a positive, becomes more negative. Or a chlorine channel opens, we're adding a negative, and that becomes more negative. It moves further away from threshold. Are we clear? Yes? So here, on this particular picture, there are multiple neurons that are targeting the postsynaptic neuron. See that? Okay. All of these neurons, presynaptic neurons, are releasing the same neurotransmitter. Is that always the case? Are all of the same neurons going to stimulate or inhibit, produce an EPSP or an IPSP to the target neuron? No, likelihood that it's like this. The more than likely that we're going to have some inhibitory neurons, IPSPs in red, and some EPSPs in green. The point of the receptive segment is to take in all of that information. Okay? And here's a little picture over here showing you a neuron that has loads of presynaptic axons terminating on it. So, how does the target postsynaptic neuron determine whether or not to produce an action potential? That's what we call summation. Right? Summation means to add together. So the initial segment, the axon hillock, its job is to add up all of these signals, some positive, some excitatory, some inhibitory, right? And determine if and when all of those signals do they ever reach threshold? Or do some, like be inhibitory, some are excitatory, back and forth, up and down. Eventually, we reach a point where threshold is met. That is called summation, right? Of the EPSPs and the IPSCs. And that happens at the axon hillock, or the initial segment. Okay? So the sum, I love this, may or may not reach threshold membrane potential. What happens if it doesn't reach threshold? What happens if we never reach threshold? There's no action potential. It just keeps adding things up, right? If we don't reach threshold, and then sodium ion channels close, and then we move sodium back out, it goes back to resting membrane potential at some point, right? Or IPSPs are added, and we return back to resting membrane potential, possibly get hyperpolarization. If we don't reach threshold, there's no action potential generated. But once we reach threshold, what happens? Once we reach that critical voltage, what happens on the initial segment? Let's back up. Nate. Which voltage gated channels? Yes, you're right. Which voltage-gated channels open first? Not calcium. Calcium. Where are calcium channels? In the axon terminus. We're at the initial segment. What type of channels are present? Sodium channels first. Okay, so the initial segment and the conductive segment have what type of channels? Voltage-gated, sodium, and potassium channels. So first, if we reach threshold at about minus 55, that triggers the opening of voltage-gated sodium channels. Now we start to generate an action potential. So those sodium channels open. What part of the action potential are we in? Depolarization. Yes? Okay, those sodium channel is going to be open until it hits what voltage? Positive 30. Okay, so summation takes all of that information, those inhibitory signals or excitatory signals, and interprets them, right? Collects them all together, moves the needle closer to threshold or further away from threshold, right? And there are two different ways that summation is conducted. Either spatially across the entire receptive segment, okay? Taking all of those signals in at once and determining yes or no. Do we reach threshold or not? Okay? Or temporally. Temporally refers to time. Okay? How often are the signals released? What happens if we have one neuron that is like releasing those signals left and right more often? That actually moves the needle closer to threshold. Does that make sense? It's going to look something like this. Here is your spatial summation. All of these neurons firing an EPSP onto the postsynaptic neuron at once. Okay? But one particular neuron isn't enough to reach threshold. But we add them together and we keep moving closer and closer to threshold, eventually reaching threshold. What's happening here? The little down part. Why isn't this just a straight increase to threshold? Why does it go up after P1? It goes up and it comes back down. And comes back down. And then P2 goes up and comes back down. Why isn't it just straight? What do we think? Like, why does it go back down? So, what's happening? Molly?
Is it because of the IPSP?
Could be, right? There could be IPSPs in there. It's a completely logical explanation, right? That and the sodium potassium pump is moving things out, right? So we're restoring, like in between impulses or excitatory potentials, the neuron is trying to reset itself, right? So that's how we move closer and closer to threshold. Okay, here is your temporal. There we go. Here's temporal. And here, one neuron is just continually firing on the target neuron, on the postsynaptic neuron. So your postsynaptic potential, all these are coming from P2, but each one is moving it closer and closer to the threshold. We add them up, eventually we reach threshold, and we produce the neuron produces an action potential. What's happened? What is going on? What's happening right here? This part of the neuron. This part of the action potential. Depolarization. What's happening over here? Repolarization. What channels are open during depolarization? Voltage-gated sodium channels. What happens right up here at positive 30? What happens to those channels? Someone besides you two.
Na shuts off and potassium opens.
Sodium channels close, potassium channels open. Does that make sense? Right? Potassium starts to, so the potassium channels open at positive 30, sodium channels close, potassium starts to leak out, and as it leaks out, positive ions, right, positive charges go out, it becomes more negative, eventually moving to getting closer to resting membrane potential and actually moving past it. But we'll talk about that in a little bit. Okay? What we've talked about, whether or not we reach threshold, is what we call the all or none law. Either we reach threshold and produce an action potential, and that impulse then is sent down the neuron, or we don't and nothing happens. Right? They use the example of like firing a gun. You have to produce enough pressure to pull the trigger, but once you do, there's no getting the bullet. The bullet fires and there's no getting it back. Okay? These action potentials are different than gradient potentials. They don't differ in intensity. It's the same intensity every time. The gradient potentials can be bigger or smaller, right, depending upon what neurotransmitters targeted, what receptors there, how often the neurotransmitters, how long the neurotransmitter stays bound to the receptor, etc. Right? That is not the case with an action potential. It's all positive 30, right, and back, or nothing at all. So, the initial segment starts the action potential. Sodium channels open, voltage-gated sodium channels open, right? We depolarize, get up to positive 30, and then that part of the membrane depolarizes and then repolarizes, and the next part of the membrane depolarizes and repolarizes, and we start to move down. Same thing happens as we move down the conductive segment or the axon, right? So your action potential, right, leads to depolarization followed by repolarization, okay? And it's propagated down the axon, down to the synaptic knob. That voltage change is carried out, what we call propagated, down the axon. Once it reaches the axon, then what happens? What's the next part? What's the next thing? Next step. So we've propagated the action potential down. Now, we open what? The voltage-gated calcium channels. And which way is calcium going to move? Into or out of the cell? In. Right? So calcium is higher outside the cell than inside the cell. Calcium moves in, binds to the synaptic vesicles, and triggers the release of neurotransmitter into the synapse of the next neuron. Okay? Questions? All right, so the next few slides are just going over the same stuff. Okay? We could talk about it like this. Action potential. All right, let's just go through it. Action potential. Axon hillock, what's happened here? It's kind of switched right? Now the axon, we're at minus 70. Now it's going to hit threshold, minus 55. And over here, it's showing you what's happening. Voltage-gated sodium channels open, they're activated. What's going to happen? That sodium is going to rush in. Right? What's going to happen in the minus 55? We've reached minus 55. Now where is it going to go? It's going to become more positive. Up to what? Positive 30. Right? And that's what we see right here. Why is that positive 30 a big one? Because that's what then, that voltage closes the sodium channels, right? We hit one of those gates. We hit the inactivation gate, it closes, and then it resets back to resting. So it closes, that positive 30 also leads to, right? So that closes, awesome. That's the end of depolarization. But now, at positive 30, that triggers those potassium channels to open, and potassium is going to leak out, returning the membrane potential back to rest. Okay? However, what you see here, what happens? What do you notice about this graph right here? Nate. It goes past resting membrane potential. Okay? Once it goes past resting membrane potential, past minus 70, that part that's now a hyperpolarized membrane potential. Okay? At minus 70, that's where the potassium channels start to close, but they're just a little slower. They take a little bit longer time to close, so that's why we're not at this, like we changed the slope of this, the steepness of this line, right? This is happening really fast, potassium's moving out, then it starts to close, and they start to kind of, not all of them are closed completely quickly, but they kind of close over time, and so that's why the slope is a little less, right? Because it's just going a little slower. Eventually, it does stop, and guess what happens? We reach back. The neuron returns back to resting membrane potential, right? That has to do with part of the sodium-potassium pump, the potassium leak channels, the sodium leak channels, and return the membrane potential back to the resting minus 70. Questions? Now that happens in adjacent parts of the membrane. First in the initial segment, then through the conductive segment, down like one part of the membrane, and that will depolarize, start to repolarize, and then the next part of the membrane will depolarize while the previous part repolarizes, and it happens just like a domino effect, one part after the other. And it leads to conduction of the action potential down the axon, and only in one direction. Why is it only in one direction? If we're depolarizing here, why is it not depolarizing backwards? Well, that's where that inactivation gate of the sodium channel comes into importance. All right? So that for that period of time, we don't go backwards because the sodium channels won't reopen, right? Even though for a period of time. That maintains a one-way conduction of the impulse from the cell body down the axon to the axon terminus. We don't go backwards. Are we clear? So here are the different parts. You should study this particular graph and be able to tell me what's happening in each part of it. All right, I basically just gave it to you over here, and we've gone over it pretty much ad nauseum here. Yes? Are we clear on this? Any questions on what this means? Okay, I'll take your silence as a yes. Apparently this is just not working today. All right? Now, in the conductive segment, there's what we call a refractory period, both absolute and relative. An absolute refractory period means that no stimulus can be initiated. You can't generate another action potential. Okay, so an action potential is generated and it's propagated from one part of the neuron, one part of the axon to the next. That original part of the axon has to reset itself before another impulse can be propagated down. Okay? At that point, the sodium channels are open and then inactivated, right? There's your inactivation gate. They're shut off, okay? And this ensures that propagation goes only in one direction. So at first, right after depolarization happens, there's the absolute refractory period. And then there's what we call a relative refractory period, okay? Happens right after the absolute refractory period. Remember that the inactivation gate only lasts for a second or a short period of time, and then the sodium channels go back to resting. They could be opened again if there's a high enough stimulus, a strong enough stimulus to overcome threshold again. Okay? Because we've had this depolarization that happened, and this part of the membrane is starting to repolarize. So repolarization happens, those positive ions are leaving the neuron, right? If we have a high enough stimulus, we have to overcome that loss of positive ions and reach threshold again in order to, right? Because that hyperpolarization that's occurring, right? So it's slightly hyperpolarized. You could generate another action potential, but it takes more stimulus to achieve threshold because it's hyperpolarized. Okay? Questions? I don't know what's going on with this thing today. Oh well. All right. Questions? No. Now, the other characteristic of the conductive segment is how it propagates the impulse. Okay? We know that the axons and neurons can be what? Myelinated or non-myelinated. All right, so we have let's undo that. Draw our little multipolar neuron here. Okay, we'll draw two of them. Lovely. Yes, two neurons. One of which is myelinated. And we'll say this is a peripheral neuron, somewhere in a nerve. So what type of cells have made that myelination? In a peripheral nerve, Schwann cells or neural lemocytes. Okay? The purpose of myelination is to increase the speed of conduction, right? The impulse goes faster. So in the non-myelinated axon, the impulse comes down the axon hillock, one part of the membrane at a time, and it travels down the entire length of the axon. Clear? Pretty straightforward, right? With a myelinated axon, the conduction is not continuous. It's what we call saltatory. It jumps from one part of the axon to the next. Typically, it's the parts that are exposed or not open or not covered by the neurolemocytes or the myelin sheath. Does that make sense? So in this case, comes down the axon hillock, jumps over to the next space, jumps, eventually gets there faster. Right? That's kind of the way you can think about it. Biochemically, that's not really what happens, right? We have the voltage gated channels are present on the exposed part of the membrane. Okay, so this part depolarizes. That depolarization actually travels through where the myelin sheath is, because there's no sodium potassium channels, right? That's covered. They're not losing ions through there. So that depolarization travels down, depolarizes the next spot, and then that travels, right, extends from one to the next, right? It effectively ends up in that kind of jumping idea, but within the cell, that biochemically, it's still happening inside the cell. Okay? So your saltatory conduction happens on myelinated axons, right? And this kind of runs down the whole part of it. And I gave you the idea of what it looks like. All right, this particular slide goes through the nuts and bolts of it. Okay? So basically, which one's faster? Saltatory or continuous? Saltatory. It's like when you're going through the airport, right? And you're walking at a regular pace, but then there's that people mover, that conveyor belt that goes a little bit faster, right? And if you walk at the same pace on that, then on the regular ground, you're going faster, right? It's kind of like that, right? Imagine if now that person that's on the conveyor belt is skipping. Now it's going really fast. All right, questions? So saltatory conduction happens in myelinated axons, and it increases the speed of conduction, right? So speed of conduction depends upon, number one, whether or not there's the axons are myelinated, okay? Myelinated axons are faster than non-myelinated axons. Second, the diameter or the thickness of the axon also increases the speed of conduction. Right? So the fastest axons or the fastest neurons are the ones that are wide or thick and myelinated. And the slowest ones are the thin ones that are non-myelinated. And we'll go through that in a little bit. This is as well. Eventually, the impulse reaches the synaptic node. The impulse then causes the opening of voltage-gated calcium channels. Calcium moves into the axon terminus, binds to synaptic vesicles, and causes the release of neurotransmitters into the synapse via exocytosis. Okay? Now, that postsynaptic neuron we've been talking about becomes the presynaptic neuron that neurotransmitter that's being released is going to target another neuron or even a muscle or something else. All right, so here's your release of neurotransmitter. Calcium ions, voltage gated, they open, trigger the release of the influx of calcium, and then the release of neurotransmitters. And that neurotransmitter then can diffuse across the synaptic cleft and bind to its receptor. So here are the next few slides are all explanations of taking in the message, right? EPSPs or IPSPs, what happens throughout, okay? So here, receptive segment, right? Sodium channel opening in the receptive segment. That is a EPSP. Opening a potassium channel is an IPSP. Opening a chlorine channel is an IPSP. right? Some excitatory, moving it closer to threshold, as you see here in the green, all right? Or an IPSP, hyperpolarizing the membrane, moving it further away from threshold, making it more difficult to reach threshold and therefore generate an action potential. Those are all collected at the initial segment, okay? And then the initial segment will help determine whether or not we reach threshold. The process of tallying all of those EPSPs and IPSPs is called summation. Summation occurs either spatially, bless you, or temporally. Spatial summation, multiple neurons releasing signals at the same time. They're collected together, reaches threshold, generates action potential. Or temporally, one neuron releases EPSPs, stimulus, eventually, well, let's say one neuron, multiple stimulus in a short period of time, eventually reaches threshold, and we get an action potential generated. Those EPSPs and IPSPs are called what? Are examples of what? What are they called? Those are called graded potentials, correct? They're graded, they're local. They could be different in magnitude, all right? They could go closer to threshold or further away. Then, on the conductive segment, we reach threshold, and now we talk about depolarization that occurs. All right, so we have this graph. So we reach threshold, sodium in. That means the sodium channels are open. Potassium out. Potassium channels are open. Sodium channels closed. Hyperpolarization, we go past resting membrane and then reach back to resting membrane potential. All right, and it shows each of that. That happens at one part of the membrane, then the next part, and the next part as it moves down and conducts the electrical impulse away from the cell body down the axon. And the propagation of that, the speed of that propagation is dependent upon whether or not that axon is myelinated. If the axon is myelinated, then we have what's called saltatory conduction. And this picture shows you how it works, right? So depolarization happens in this part of the membrane, okay? And then that depolarization travels under where the myelin sheath is and then works to depolarize the next part of the membrane at the neurofibril nodes or the nodes of Ranvier. All right, so we effectively have the saltatory conduction that occurs, jumps from one area to the next. And saltatory conduction is faster than continuous conduction. Finally, we reach the transmissive segment. That electrical impulse comes down, triggers the opening of what? What channels open in the transmissive segment? Voltage-gated calcium channels. All right? Calcium moves into the cell and triggers the release of neurotransmitters from the synapse or from the axon terminus, right, via exocytosis. Then the neurotransmitter is released into the synapse, diffuses across the synapse, binds to its receptor, and then the process starts again on the targeted neuron. Maybe this is not the target, and maybe this is not a neuron, maybe this is a muscle cell, maybe this is a gland, whatever it might be, right? But we're releasing the neurotransmitter on the target cell. Now, I've told you a difference between graded potentials and local potentials. Graded and action potentials. Graded potentials are local potentials. They are present in one area. An action potential goes from initial segment all the way down the conductive segment to the axon, so it travels, all right? Graded potentials only happen in the receptive segment, right? Because due to ion flow, due to the types of channels that are present in the receptive segment, right? Those graded potentials can be negative or positive, right? They can move closer to threshold or they can move the needle further away, right? And they are graded. They can be different magnitudes, right? They could be a little bit bigger, a little bit smaller, depending upon what we're talking about. All right? That's a stark difference from an action potential. An action potential only happens in the initial segment and down the conductive segment. And that is the action potential is a result of sodium moving into the cell during depolarization and potassium moving out of the cell during repolarization, right? And that is dependent on your all or none law. There's no grading, right? There's no grading. Sodium channels close at positive 30. They stay open from minus 55 to positive 30. That does not change. Are we clear? So there's no, action potential of plus 15. No. Does that make sense? It goes minus 55 to positive 30, and that's when the sodium channels close and potassium channels open. All right, and then repolarization happens while potassium moves out of the cell. All right, and that propagates down the entire axon to the synaptic knob. All right, so there's a difference between graded and action potentials. You should understand the difference. Graded potentials are IPSPs and EPSPs. Okay? Action potentials are propagation down the axon. And I've talked about conduction speed. Okay, so first, conduction speed depends upon whether or not myelination is present. If it's myelinated, it's faster than if it's non-myelinated. And second, the thicker fibers conduct faster than the thin ones. Thicker, right? There's more movement of ions. Things can move a little bit easier, right? There's less resistance to current flow. So less resistance, remember Ohm's law, voltage, current, resistance. Less resistance means current can move faster. Therefore, thicker segment, thicker diameter, easier current change, easier flow, faster conduction. All right, so the thicker the fiber, whether or not it's myelinated, those are the fastest conduction that happens. Okay, so based upon that, we can divide fibers or neurons into a couple different groups, which are characterized by whether or not they're myelinated, how thick the axon is. All right, so group A has a conduction velocity of about 150 meters per second. Okay, these are your fast fibers. Large diameter, myelinated fibers, they happen the fastest. Think about what they are. So group A here are most somatic sensory neurons. Somatic sense, how fast does light information hit your eyes, gets to your occipital lobe of your brain, is processed real quick, right? So does sound. So does a lot of your other sensory information. How fast do you respond to that? Think about how fast you respond, a muscle response to your eyes. You see me walking back and forth, right? Think about how fast the response is of your motor neurons. Motor neurons are also large diameter myelinated fibers. Okay? Group B conducts at 15 meters per second. Okay? And then group C at 1 meter per second. These two are smaller diameter, and then group B are the smaller diameter myelinated. Group C are the smaller diameter unmyelinated. So group C are the slowest conduction. Okay? Some visceral neurons. How long does it take to recognize whether or not you're hungry? Oh, visceral neurons, movements, right? How do things move around in your organs? Some somatic sensory neurons from the skin, right? A little bit slower than what you see from your eyes, right? The sensory information from your eyes and ears. So different types, again, depends upon presence of myelination and the thickness of the fiber itself. Okay? And then finally, your impulse frequency. Action potentials can occur pretty fast. Their amplitude or the size of the action potential does not change. Positive 30 all the time, all or none, right? So the amplitude is constant, but the firing frequency varies with stimulus strength, right? So how fast or how often an action potential is generated depends upon the signal that starts. Example, bright lights. They cause faster firing frequency on the optic nerve than dim lights, right? And in response, what does your body do? It opens the pupil, right? Dilates the pupil, etc. Okay? When motor nerves fire faster frequency, it causes muscle to generate more tension, all right? For neurons that can use different transmitters, because they can. We'll talk about neurotransmitters on Monday, right? And the different ones. We'll talk about acetylcholine, dopamine, right? And adrenaline or norepinephrine, epinephrine. The firing frequency can influence the type of neurotransmitter released. Now we're talking about how things change. We're up to now, we've talked about motor neurons, acetylcholine as our only neurotransmitter. We haven't talked about, well, how do neurons control what the response signal is on the postsynaptic neuron, right? So if we have a neuron that can release both acetylcholine and norepinephrine or dopamine, right? A couple different neurotransmitters. Which neurotransmitter is released from that neuron can be determined by how often that action potential comes down the neuron. The more means more faster it happens, maybe it releases adrenaline or epinephrine, right? Slower it happens, maybe it releases acetylcholine, back and forth, as in examples, right? So it's just the ways to control the signal that's being released. Questions on action potentials, summation, graded potentials versus action potential. Before you go, I have a Socrative quiz for you to complete. Answer the questions based upon the lecture